Well, this session called Life Science actually, this morning we already have 2 sessions, also Life Science, anyway. And originally we have 10 speakers, but now reduced to 9, unfortunately, and Aaron Shihalov is not be able to come. So 9 speakers, and we start half an hour later. So this afternoon, we try to keep time on, okay? And I'm not going to make longer instruction, because if you want to know who's who, just check this book list, okay? And first speaker is Professor Dai Hongjie, 2017 NIH Director, Pioneer Award winner. Yes, welcome.
Okay, good afternoon. Very nice to be here to tell you a vision that I have. Yeah, so basically I want to tell you an imaging technique that I feel can make medicine visualizable. So basically you can see medicine work in real time. And so the question we have in hand is, can surgeons visualize cancer cells, right, when they are doing a surgery to remove a tumor? Okay. And can you visualize Alzheimer's disease plaque in the brain? And when you do ADC drug, antibody drug complex delivery, are these drugs actually reaching the tumor? Right? Can you see them? Can you visualize them? How do you know the drug is getting to where you want? And I mean, the question goes on and on in medicine. Right? In CAR T immunotherapy, are the T cells actually going to the tumor to kill the cancer cells? How do we know? So this is this morning there was a discussion about pain relief, right, for ion channels. How do you know these drugs are getting to the ion channels in the body, and the body is very complex. So I believe that visualization in the body at cellular level or molecular level is a frontier of medicine, okay, at very high resolution, and be able to see everything in real time. And this is limiting. We don't have such capability. Even in immunotherapy, whether T cells are responding, whether they are getting to, CTLs are reaching to tumor to kill cancer, it's not all that clear. It's trial and error. So we are developing a technique called infrared fluorescence imaging. I mean, everyone here knows about fluorescence imaging, but we are pushing the fluorescence to infrared range, okay, to develop the so-called infrared vision, because infrared vision gives you penetration depth. So we want to be able to see things in the body to see if they get to the targets, okay, and see if treatment is working, if every cancer cell is removed by the surgeon. And we are doing this by combining chemistry, material science, and integrating with robotics and artificial intelligence, because quite often you want to target something, you need to have a very good ligand. And those ligands binds to specific proteins, and that needs to be optimized by AI. So the journey started in 2009 when I'm a physicist by training, so we studied carbon nanotubes at the time. So we decided to inject a little bit of a carbon nanotube in the mouse, and then we start to see these nanotubes circulating in the body of a mouse, and we realized that because we're imaging the infrared range, it's 1,000-some nanometers, we actually can see through the skin and look at the blood flow in the body, non-invasively. We don't have to do any surgery to open it up. And the physics here is very simple. Light scattering scales with lambda to the minus alpha power. So if you go to long wavelength, you can get scattering to be reduced. This is exactly the physics of light scattering by fog, okay? You cannot visualize because light is scattered. So we showed that in a mouse model, if you image in the infrared, you can actually see blood vessels in the leg of a mouse very clearly because you cleared up the fog. You reduce the scattering. Okay, so this is a very large difference in imaging. And the physics, again, is scattering is going down as you increase the wavelength of light. So here is a movie of a mouse head. We can actually do infrared fluorescence imaging and penetrating through the skull and the scalp of a mouse and directly looking into the mouse cerebral vasculature, non-invasively. We didn't open up the mouse or anything. So we have such infrared vision going into the body. So a frontier in this area has been combining chemistry and nanoscience to make fluorophores that emitting in the infrared. And there is many classes of emitters like carbon nanotubes, quantum dots, and molecules. Okay, there are many people working in this field trying to get emitters in the infrared. And then also you can do chemistry or biochemistry to link ligands or antibodies or peptides or drugs to these probes so that you can see what you want to see in your body. So you can target certain things in the body and you can image them very specifically. So we've done this with tumors, okay? And we have done this for T cells. We can image T cells and cancer cells looking at immunotherapy. So right now, at this stage, we're beginning to do clinical translation, trying to bring this technology to the clinic. Okay, so for surgeons to see blood perfusion better, cutting diseased tissue better, and also we're integrating with robotics so that we can have robotic surgery guided by these fluorescence imaging. And we are cutting tumors cell by cell until all the cancer cells are removed. Okay, we can have very high precision to do that. And we're developing imaging instruments and a series of probes for cancer, for neuroscience, and so on. So the answer to the question I raised in the beginning is yes. Thank you.
Next speaker, and Francis Ulrich Hadi. Welcome.
Thank you. It's a pleasure to talk with you about some of our research on how cells make functional proteins, the process of protein folding and its implication in various diseases, mostly neurodegenerative diseases. I should say that I'm a huge fan of curiosity-driven research. Not only do I think that that's the most exciting way to do research, but history shows, of course, that it is the driver of major advances in medicine and technology. Now, proteins doesn't seem to go forward. Now it's working. Proteins are the workhorses in our cells. They do practically every cellular function. They come in many shapes and sizes. There are approximately 20,000 different proteins in a human cell, some in only a few copies, some in millions of copies per cell. I have been fascinated with the question how cells achieve their final shape, their fold, and is there perhaps a machinery that helps them to do so? And the answer to this question is yes. We have molecular chaperones, a set of dedicated proteins that help other newly made proteins to get into shape. Now, the protein chain is made on the ribosome. We heard about this as from amino acid building blocks. The ribosome, you could view it as a molecular typewriter. There are 20 amino acids. Each type of protein has a specific amino acid sequence. This information is stored in the gene, and for the ribosome to read it, it must be transcribed into mRNA. Now, importantly, the amino acid chain has to fold into a unique three-dimensional pattern in order to become functionally active. Initially, this process was thought to be a spontaneous reaction, not requiring any further machinery, but we now understand that molecular chaperones are required by many proteins in order to fold efficiently, and many proteins, in fact, critical proteins like actin, tubulin, require this machinery to fold at all. Now, in the presence of the chaperone system, a protein will fold properly into its functional state, just like this beautiful origami bird, whereas in the absence of chaperones, proteins misfold, and because misfolded proteins are sticky, the chains will stick together, will form clumps, aggregates, just like origami gone wrong. Now, importantly, these protein aggregates are often toxic to cells and are associated with various diseases. I'm showing you here how the chaperone system can avoid this from happening using the HSP60 as one of the best studied systems. The HSP60 is a container. It receives unfolded protein from another chaperone called HSP70. The client protein binds into the central cavity of HSP60 and is then encapsulated by a lid-shaped cofactor, a really fascinating reaction. It is now allowed to fold in isolation because there is only a single chain of amino acid. It cannot clump together with other proteins. And after a couple of seconds, the lid will open and release the folded protein. Now, it turns out that this function of preventing protein aggregation is medically very important, because when chaperone systems fail, this can facilitate the formation, the manifestation of various age-dependent diseases such as Alzheimer's disease, Parkinson's, Huntington's disease, which are associated with the formation of protein aggregates. Here is how such an aggregate looks like. We obtained this image by cryo-electron tomography in collaboration with the Baumeister group at my institute. The aggregates are seen in blue like a disordered array of spaghettis, and these aggregate deposits, as we could show, sequester various endogenous cellular proteins, and they also displace cellular structures. These are reasons why these deposits are actually toxic to cells, especially to neuronal cells, which have a very complex architecture. Why is this happening? This is happening apparently because during aging, the cellular capacity of protein quality control, especially of molecular chaperones and other factors, undergoes a decline. And this results in an imbalance between the production of misfolded protein and the available capacity to prevent it. Our goal is to rebalance the system. We would like to find ways to manipulate cellular quality control machinery to set it back into a more useful state, and hopefully this will then allow us to develop new strategies for the treatment of diseases like Alzheimer's and Parkinson's disease. I would like to appeal to our funding agencies to continue funding basic curiosity-driven research. Thank you so much.
Next speaker is Professor Dr. Liu Nianxing. He's the president of the first affiliate hospital of University of Science and Technology of China, and also called Anhui Provincial Hospital. Welcome.
dear colleagues. As a surgeon, today's topic is analysis of the tumor immunobarrier in primary liver cancer. In this case, it's primary liver cancers, mainly is the hepatocellular carcinoma. And as we all know, the incidence and mortality rates of the primary liver cancer ranks 7th and 4th, respectively, but it's the 2nd and the 4th in mainland of China. And the primary liver cancer, there's 4 treatment challenge. The first is low rate of resectability at the initial diagnosis. For most of them, is unresectable. And the second is the high post-operative recurrence with no standard adjuvant therapy. And the third is limited beneficials from the immunotherapy. And the fourth is lack of the predictive models for recurrence and treatment practice. Today's topic is focused on the third and fourth. This is a precise mapping of the tumor ecosystem. Cellular and atlas is essential. And we identified the tumor immunobarrier structure characterization. And as a surgeon, we have the chance to get the samples from the patients who do the conversion therapy before operations. There's a response on non-response. For these 2 groups of the patients, we get the samples. We identified the tumor immunobarrier, a barrier structure formed through the colonization of the abundant SPP1 positive macrophage and cancer-associated fibroblast was observed as a tumor margin in patients for these unresponsible patients. And we analysis of this single-cell RNA sequence and the TCGA database consistently demonstrate elevated levels of the SPP1 positive macrophage in tumor tissues related to the normal tissues. So we identify these structures. But these structures, not only the SPP1 positive macrophage, it's combined with the cancer-associated fibroblast. They form the tumor immunobarrier. And the tumor barrier restriction of the immunocell, especially for T cells, infiltration into the center of the tumor. So after this, we do the multiple tumor models revealed that inhibiting the SPP1 positive through the macrophage-specific deletion or target intervention suppress tumor growth and have increased the capability of the immunotherapy. And this is what we had done for how to increase the response for the immunotherapy. But for the recurrence, it's also the high risk for the patients without lung longevity. So we do the spatial immunoscore system predict hepatocellular carcinoma recurrence as a cover paper published in Nature. And with samples with the spatial information were obtained from the cohort of the 17 patients. We trained the NI models that identify 5 genes, at last 5. As the first is 241 genes with strong links to the hepatocellular carcinoma recurrence. These genes was subsequently used to construct, we call this model TIMES model, we call TIMES score. And validation was performed using 3D bioprint and genetic knockout mouse models and data from the multi-center analysis. So for non-recurrence patients, the invasive front regions is enriched with measured CD3 negative and CD57 positive mature kill NK cells. For this recurrence, mature NK cells are less abundant at the invasive front. So we analyze these 2 different groups. We identified 241 genes, but this was not be used in clinical work. It's too huge gene we want to test. So we analyze these genes to 23 genes, but it's also too large. So we 5 candidate genes for predicting ACC recurrence was in our group. We used machine learning. These 5 genes were combined with optimized white values. We called these models we call TIMES model, and these courses have the superior accuracy and higher specificity. The AUC is 0.82. It's the highest in the world exists for predict the recurrence. So as this SPON2 positive NK cells represent an active anti-tumor NK cell subset, we found these NK cells in ex vivo tumors models. These overexpressed NK cells exhibiting higher mortality, but if we knock out NK cells, losses maturation capabilities for these tumors. So these NK cells with active the T cells in the immunomacro environment. So we launched a free open source online tools has been launched to support upload all over the world to analyze of the post-operative immunohistochemistry image or protein codification results. There's already have 35 countries visit this website. Welcome all of you to visit this. Thank you for your attention.
Next speaker is Professor Barton Roska, and 2024 Wolf Prize in Medicine winner.
Vision is a process that starts with a video that enters your eyes, it's captured by the retina, and then it marches to higher brain centers to form a percept. In the last 30 years, I've been studying the mechanism of vision at the level of cell types and circuits and pathways, how these interact to yield a percept. In around 20 years, inspired by the very detailed understanding we have about retinal circuits and molecules and cells, and about the fact that for most visual diseases, we have absolutely no therapy. And the third point, that most visual diseases originate in the retina that we cannot treat, we started to get interested of developing new therapies. Indeed, this is a survey from the US a few years ago when they asked people that ranked the worst conditions among the one that they received. And blindness ranked number one, probably for the reason that we are entirely addicted to our cell phones and computers. It takes over our social and other parts of our life. Now, if you ask yourself, what is the biggest blinding disease that will dominate in the next years, there's a surprising answer to that, and that is nearsightedness, called myopia. So myopia has been around 20% 60 years ago, and in China and in Asia, it's around 95% among kids. And what happens is that eye grows very large, that pulls the retina, creating so-called myopic macular degeneration, myopic glaucoma, and retinal detachment that will dominate blindness in the next years to come. Now, as you can see here, that today we have around 400 million people with high myopia, above 6 dioptry, and that will be around 1 billion by 2050. And that's something we would like to fight. And there are two ways to fight blindness. One is to use technology to restore vision in blind patients using gene therapies and light sensors that we can deliver them. So in the last 20 years, I've been developing the so-called optogenetic cell type targeted therapy in the eye with a single injection. These viral vectors find the right cell types. We can engineer them to make a sensor in the cell type we want in the human eye. And here's our first patient who has been blind since 10 years. And now you can see as he perceives an object on a table, he can locate it and point at it almost exactly where is the location. Here's another patient, also from Paris. We can even do better. Here's a pen on the table, and she can very precisely point at an object. Now, where we are today with our cohort, the first clinical trial, that these patients are quite good on finding objects, but they don't recognize faces and they cannot read. At the end of this year, we are coming to the next clinical trial, our optogenetic 2.0, when we try to restore high-resolution vision by learning from these patients and redesigning the technology and understanding what are the limitations of this. Finally, I like to say that, of course, at the end of the day, we would like not to have blind people. So one of the big challenges, which is much harder than restoring vision to blind patients, is to stop certain pathological processes. And the last 8 years in my institute, my laboratory has been occupied to understand how myopia develops at the cell types and molecular pathway level. And now we have a very, very good understanding of how the eye grows. All the molecules and cell types sort of show up in our screens made in a number of organisms, including humans. And in the last 2 years, we have found molecular pathways we can modulate. We can completely stop eye growth, and hoping that in the next few years, we are able to make some medicine that can combat this disease as well. So with this, I'd like to thank you very much for your attention.
Thank you very much. And, well, we went smoothly, yeah. And next speaker is Evan Hosko. Welcome. He's the laureate of 2004 Nobel Prize in Chemistry.
Well, thank you very much for inviting me here. I noticed that the subtitle of this session was, End of Disease. So I thought maybe that's an occasion to tell what do I think, how can we get to the end of disease, or maybe to get close to it. So I don't think we'll get to end all diseases, but we may end some diseases, provided that we'll have a good support to basic research. And as you know, there is a danger now to basic research. There are governments in certain countries, certain people that want to cut down, or they're cutting down basic research, and I think that's a big danger to human health, because basic research is the basis of all applied research that we have, at least in biomedical sciences, also in other places. And as you see in the first slide, I wanted to reiterate the importance of basic and applied research in society. So we need applied research because it benefits society directly. However, applied research and development are always based on discoveries in basic research. And to many people here in this audience, that's clear, but to some others it may not be, especially people who are connected to government decisions. So it's very important to point out that the history of science, and the medical science in particular, of which I am familiar, shows that almost every big advance in medical treatment was based on prior discoveries in basic research. That's true in the discovery of antibiotics that started by a chain observation. That's true in genetic engineering, which now many things are being done, which started by a curiosity-based discovery of the double helix and the genetic code in the 1950s, 1960s. So if we want to have good applied research, we need to invest in basic research, because that is the basis. And the basic research that is performed at universities also educates young scientists and thus provides educated scientists for applied research. So that's another reason to support basic research. But basic research is not supported by the industry. It has to be supported by governments, and some by philanthropy, but mostly by government. And governments should support basic research, because applied research can be supported by the industry. But the benefits of the investment of society in basic research are eventually returned to society. So that's important to remember. And to show why I was convinced of that, I was convinced because my own research on how proteins are degraded in cells. So that was a subject, a basic research that interested me for a long time. And it was known, as you heard from Dr. Hartl, that proteins are important. We have many thousands of different proteins, and they carry out almost all the functions in our cells. But proteins can get bad, they can get unfolded, misfolded, and then they are toxic. Or proteins can just finish their function, and then they have to be removed. So they are removed by protein degradation, and certain proteins have to be eliminated at certain times, so the system is highly specific and highly regulated. And our question was, what is the system that degrades proteins in such a high specificity and sophistication? And together with my colleague Erwin Rose and with my then graduate student Aaron Chehanover, we discovered the system. It is called the ubiquitin system. So proteins have to be marked for degradation. A certain protein that has to be degraded is first linked to a small protein called ubiquitin, and then proteins that are linked to ubiquitin are recognized by a degrading machinery called the proteasome, which degrades only proteins that are marked by linkage to ubiquitin, and so the protein is destroyed and ubiquitin can be recycled and used again. So this is in a nutshell a discovery. It was a discovery in basic science. We never thought about any applications. We didn't want to cure anything. We just wanted to satisfy our curiosity. But that eventually led to the development of new drugs because it work by other people showed that the ubiquitin system is important in almost every basic biological process in the cell, and that abnormalities in the ubiquitin system can cause diseases such as major diseases such as cancer or neurodegeneration. And because of that, drug companies began to develop new drugs based on our discoveries targeted against the ubiquitin system. The first drug was a proteasome inhibitor called bortezomib or Velcade, which turned out to be highly efficient in the treatment of multiple myeloma, a bone marrow cancer. It selectively kills bone marrow cancer cells at very low concentrations, so it is a highly targeted drug against this disease, and actually it benefited greatly the people in this disease. It made, together with some other drugs, a revolution in the treatment of this awful disease and allowed people to live more normal health, normal type healthy life. So that is an example of how basic research eventually leads to important applications, and it gave me great satisfaction that our basic research actually ended in good applications. And so our conclusion is that basic research is the long-range investment of society for technological innovation. And if we want to have highly original innovation in industry, society should support high-quality basic research. And I hope that this message will get back to the decision makers about support of basic research. So about the end of disease, I do not know whether we shall have the end of disease or when, but I do know that we will not have the end of disease if we will not have adequate support of basic research. Thank you very much for your attention.
Yeah, ladies and gentlemen, good afternoon. It's my great honor to be here to have a presentation. And today I'd like to introduce a little bit about the early screening for gastric cancer based on AI. It's well known that gastric cancer is very common in China. Oh, sorry. Second one. Yes, this is okay. Gastric cancer is one of the 4 major high incidence tumor in China. The incidence rate ranks second nationwide, and the individual with gastric cancer account for approximately 50% of the global total. However, the 5-year survival rate in China is approximately 35.1%, which is much lower than 64.6% in Japan and 71.5% in South Korea. What's the reason causes this difference among the three countries? It is early detection rate. You know, it is really low in China, only 20%, and 60 to 70% in Korea and Japan. So how can we do to improve the early detection rate of the gastric cancer? In Japan and Korea, you know, the individuals aged over 50 years old are advised to undergo a gastroscopy every 2 to 3 years. But it's impossible for us because, you know, in China we have so tremendous populations and we have shortage of the devices and experts. So by now, we only wait for us to do only by 2 steps. The first step, we should find the risk fat populations and then ask the individuals with the high risk to undergo gastroscopy. So the standard methods in China is the questionnaire methods, but the concentration rate of the high-risk individual based on the questionnaire methods was only 22.70%, and the cancer detection rate was only 1.16%. So it is methods with very high input and very low output. And recently, my group built up a model. We called it the GRAB model. It is an AI-based non-contrast CT scan model. How did I do? Firstly, 105,000 subjects with non-contrast abdominal CT scan in 18 centers nationwide from 2010 to 2024 have been enrolled. And then my group worked together with Damo Lab from Alibaba company to build up the GRAB model. And then we started to do the clinical validation and application value of the GRAB model. In internal validation cohort included over 1,000 subjects from 2 centers in China. The GRAB model achieved AUC value of 0.97. And in extra validation cohort included over 18,000 subjects from 16 centers in China, and the GRAB model achieved AUC value of 0.927. Finally, we have done the retrospective study among over 78,000 non-contrast CT scan subjects. The GRAB model successful screening 6.2% as high-risk population for gastric cancer. Follow-up gastroscopy confirmed 289 gastric cancer cases. So this work had been published in the Nature Medicine last year. Now we have started to do a clinical trial, AI-based large-scale opportunistic screening for gastric cancer. And with the non-contrast CT imaging, a multi-center prospective clinical trials. And the primary endpoint of this clinical trial is the detection rate of the gastric cancer. And second is the rate of the sensitive and specific. And by now, we have finished the employment of the GRAB model in 15 hospitals in China. And by the end of the 2028, the initial 5 centers utilizing the GRAB model have conducted screening for over 150,000 individuals. The GRAB model identified 2,073 high-risk cases, from which 1,139 individuals underwent subsequent gastrectomy. Finally, 377 gastric cancers were identified, including 106 newly diagnoses. So it is the first time that the combination of AI and the non-contrast CT scan was proven to be feasible in the screening of gastric cancer. And the non-contrast CT scan has advantages of low radiation dose, no contrast agent risk, and a wide application range, providing an effective tool for large-scale opportunistic gastric cancer screening. Thank you very much for attention. Thank you.
Okay, thank you. Next speaker is Michael Shepard, and 2019 NASCA D. Baker Clinical Medical Research Award winner.
Recently, the CEOs of NVIDIA and Lilly made a deal to join forces with NVIDIA AI and Eli Lilly know-how in drug discovery and drug development. I think this is a very big deal, and it really shows that the pharmaceutical industry is going big on AI. My talk, I'm going to try to give a couple of examples of how AI can be applied to drug discovery in cancer. Among the problems that we have to deal with in cancer is the occurrence of resistant cancer cells. Most patients probably respond to a first course of therapy, but the cancer will relapse. And in some cases, of course, it might relapse as a result of several different kinds of therapy. And so the challenge has been for a very long time to try and develop therapies that can keep up with and even get ahead of the cancer. So the origin of resistance to therapy is multifold. In this case, I'm just showing you a display of all the cancer cells of the proteins inside a cancer cell. On the top, this is a cancer cell that's not been treated, and on the bottom is another display of more than 3,000 proteins in the resistant cancer cell. Very difficult to pick out the resistance protein in the resistant cancer cell. This is where AI can lend a hand. AI can screen all of these candidates on both the top and the bottom panel and pick out one or more candidates for the resistance protein. Now, therapeutic antibodies are made formulaic, and using AlphaFold and other kinds of programs, all of which involve AI, we can actually model almost exactly what that antibody should look like if it's going to bind to the target protein and neutralize its activity, making the cancer cell sensitive again to therapy. It's amazing. When we made the first humanized antibody for solid tumors in about 1990, it took us 2 years I'm just making the antibody and showing that it worked. But now we can do this in a few months. This is a one continuous stream. Detection of the protein, identification of the protein, and then creating an antibody that specifically binds to and potentially neutralizes the resistance protein. Another area where AI has recently made it just a completely mind-bending change in the way that we think about cancer therapy is with CAR T cells. Now, everybody probably knows what a CAR T cell is. It's an engineered T cell originally derived from the patient and programmed in the lab to attack a protein target on the surface of the cancer cell, and then the T cell is stimulated to release a toxic reaction to the cancer cell. This process, we have to recover T cells from the patient, engineer them, we have to grow them up. The patient, in order to accept this graft of CAR T cells, has to undergo intensive chemotherapy, very unfriendly kind of thing, and then infused by the engineered T cells originating from the patient. That's also a problem because these T cells have been growing in vitro very rapidly. They're producing all kinds of cytokines and things that can form symptoms very similar to septic shock. There are modalities available now that will lessen the risk of this, but it's a very unpleasant thing. So one course of this kind of therapy costs between $500,000 and $1 million, serious side effects. It can only be done at a few hospitals, so you're never going to, in a single hospital, no matter how big your hospital is, you can never treat more than maybe 100 patients a year this way. So this is a problem. It's just not going to be available to most people. However, in the last couple of years, maybe only the last year, several laboratories, and especially one that I'm more familiar with at Exuma Bio, which has its offices in West Palm Beach. They work very hard given that they work in West Palm Beach. But anyway, they've been very successful at creating a new way to make CAR T cells. They've developed a kind of vector, a viral vector that they can use to target the T cells. They inject the vector directly into the patient. The vector binds to and puts the CAR machinery inside the T cell. Then the T cell increases in number. So long as there are targets there, it will increase in number. But there will be no toxicity like there is when you reinfuse the old type of CAR T cell. This is huge because there's another way, some of the ways in which AI helps here, probably the most important one is that you can make, because you're using lentiviral vectors, you can make, if there's a resistance protein, you can make another CAR and inject that until you get to the point where the tumor hopefully is overcome altogether. This whole process, it's about 10 times, it's at least 10 times cheaper than what we're doing now. The drug can be delivered in a doctor's office. You don't have to go to MD Anderson to get your drug. It can be shipped to the doctor's office and the doctor can just inject it. The other part of the AI engineering is This group has engineered what's called drivers. And in this sense, what you do is you put in a synthetic growth factor that stimulates the growth of the CAR T cells if their number gets too low in the blood of the patient. This also means that this technology can be used probably against solid tumors. So thank you very much for your attention.
Okay, ladies and gentlemen, good afternoon. Today I'm going to talk about the xenotransplantation, new avenue for liver transplantation. So the organ shortage is a global challenge. In China, organ supply and demand ratio is about 1:30. So to find a new donor resource is very important. For xenograft from the gene engineered pig is very important. So they have the 4 key advantages: technology maturity, no cost, and by safety, and few ethical concerns. So we collaborate with Tongjiang Wei and develop 10 genes edited pig, 3 genes for rejection, knockout, and 3 genes lock-in for complement regulation, and 3 genes for anti-coagulation, and 1 gene for anti-inflammatory. So as may all you know, in the United States, in Johns Hopkins, they performed 2 cases of heart xenotransplantation in living patient, and in New York University and Harvard Medical School, they performed 5 kidney xenotransplantations. So in our center, we performed the first case of liver xenotransplantation in living patient. This is a HCC patient, small, left lobe is very small, too small to support life, and also, although we get the TACE and target therapy, the tumor not shrink instead of the rupture in situ. So based on the compassionate use principle, we performed this surgery, and we just resected the right lobe of the liver and transplant auxiliary the pig liver. So you can see this is a biosecretion, and day 16, the biosecretion volume is 400 mL, and they get the very normal function. So this is a bile acid and albumin assessment. We can detect both human and porcine bile acid and albumin in recipient blood. And also, in day 10 and day 25, we do the biopsy. We cannot detect any rejection, and this is a POD21 patient. So 30 days after surgery, we got the TMA as kind of a very bad barrier for xenotransplantation. And so for this case, we asked a question whether we can get rid of the TMA. So this time we used the prophylactic, the C5b9 antibody. So it's the same way to do the tri-segmental right hepatic resection and transplant the porcine liver. And this time you can see there's no complement expression, and there's no TMA. So this is a bridging to regeneration. So we can see, okay, both in case 2 and case 1, the remaining liver gets a regeneration. And this is a long-term multi-omics of liver xenotransplantation. You can see from this chart, we can get the different time point for get the sample from the cells and tissues, and in the like single cell level and the spatial level and the protein level. So from this analysis, we can see in the liver, in the porcine liver, we got the recipient Kupffer cell, macrophage cell, infiltrated into the graft liver, and the macrophage in the porcine liver gradually disappeared in both cases. And this infiltrated human macrophage create a pro-thrombotic leach through the VEGF-centric pathway. In case 1, you can see the complement activation induced the endothelial cell injury, leading high VWF level expression in endothelial cells and cause the TMA. But the case 2, we use the prophylactic use C5b9 antibody. There's no endothelial cell injury and no TMA. So from the spatial analysis data, we can see, we can detect the human SPP1 positive macrophage and the porcine LOX positive substate in this liver, and formed a kind of the cross-species regeneration leach. So this is the key finding. I think no hyperacute and acute rejection in early stage. And we identification of TMA as a critical barrier, and the TMA is prophylactic control with the C5b9 antibody. And auxiliary liver xenotransplantation can bridge to native liver regeneration. Thanks very much for your attention.
Okay, it's my turn as the last speaker. Thank you. Dear colleagues and friends, my talk is Packaging and the Universal Health, from entry, transcription, replication, and assembly to the drug discovery. Since the dawn of the human civilization, infectious disease has always been a major challenge threatening human health and social development. For this, we have learned a lot from COVID-19 pandemic. Based on the current global health landscape, the complexity of risk has significantly increased. Challenge at the levels of key technology, prevention, and control have become even more crucial. Most of pathogens that caused several global diseases, severe global diseases, lack of effective prevention or treatment method. This PPT shows list the 25 most burdensome pathogens globally. But you can see that in red, there are 20 of them lack of prevention or treatment method. At present, there is an urgent need to develop a series of high-effective vaccine antibodies and small molecule drugs for prevention and treatment. We are very pleased to see that there are many breakthroughs have been made since COVID-19 pandemics. New cutting-edge technologies keep emerging, especially AI, everybody mentioned AI. Convergence with biotechnology-driven innovations in drug discovery. Prevention and control of X diseases has been elevated to the nation and the global strategies. The major economies such as WHO, USA, European Union, China, have all clearly defined the goals for dealing with pandemic infectious disease and have formulated specific action plans. Well, in the past 3 decades, we have been exploring field of pathogens of emerging, re-emerging infectious disease, hoping to answer major scientific questions and cure the disease. We have solved more than 30 important virus particle structure and many virus compartments at atomic resolution. They are including flu, COVID, flavivirus, picornavirus, hepatitis B and hepatitis A virus, herpes virus, Afghan swine fever virus, as well as a number of membrane protein complexes from Mycobacteria tuberculosis. Since 1995, we have published over 480 research papers, among which 32 papers were published in Nature Cell Science. Our team has made a significant discovery in pathogens entry, replication, transcription, and assembly. Based on this finding, several important targets has been provided for vaccine, antibody, and small molecule drug design. 2019 WAA summit in Shanghai, I presented the structure of Afghan swine fever virus at the summit. We revealed its 5-layer architecture, which is the largest virus particle has been solved so far. In doing so, we identified the composition of various proteins and pinpointed key epitopes, shedding light to show virus assembly itself. Furthermore, based on this finding, we have developed a recombinant nanoparticle vaccine against Afghan swine fever. This vaccine has been approved for clinical trial by Chinese Ministry of Agriculture last year. A recent breakthrough is the architecture of hepatitis B virus as particles. We have worked out the structure of HBs and antigen membrane envelope particle. This gives us a clear picture how virus envelope is built, how virus takes shape. This information is super helpful for the developing better antivirus drug and vaccines. Of course, this is one of my dream work in 3 decades. Since the last 10 years, we are systematically working on the TB structure of key drug target complexes by focus on the critical pathway, including the cell wall biosynthesis, energy, metabolism, transmembrane transport systems, and pathogen and host interactions. We have made a series of significant breakthroughs in the structure of TB drug targets, which have made a reasonable big impact in the field. This is the result of our team's long-term dedication and joint effort. I would like to express my gratitude to all my students, all my colleagues, and all the collaborators. I'm extremely proud and honored to work with them. Thank you. Okay, at this point, as a moderator, I would like to conclude this session. Let's express our gratitude to all the speakers. We also hope this integration of basic medicine and clinical medicine will enable humanity to overcome diseases and enjoy long and healthy life. Thank you.